So today I'm going to be teaching you guys about how Ebola can be neutralized by an siRNA drug. And so just to start off, I'll ask you guys some questions, see what you know. So what do you guys know already about viruses?
It can be spread.
Alright, do you know how they work?
Is it when they change, like they mutate the cells?
So just viruses in general are made up of nucleic acid. They have a coat of protein, and some of them have lipid membranes. It doesn't really change that much about them. And then each virus only has one or two enzymes, and for this reason they need more enzymes to do their bidding basically, and so they use the enzymes in our cells. And then the lytic cycle is the cycle of how a virus spreads through the body. So first it takes over a host cell, and then it uses this host cell's enzymes to perform its functions to create more. And then once it's done, the cell is destroyed and the new viruses explode out and go into other cells, and the process just repeats. Ebola in particular is very deadly. It has mortality rates of up to 90% in some cases. Outbreaks of this have caused many casualties. So Ebola is more effective than other viruses for several reasons, and one of them is because in the cells there's something called tephrin. This is a protein that normally affects the viruses and how they replicate, and normally when it's working it prevents the virus from leaving the cell. Like all the viruses that are made in the cell, it prevents them from leaving, and Ebola has developed a way to disable this. siRNA, this stands for a small interfering RNA. They're highly specific and very potent drugs. They are used to infiltrate infected cells and alter the virus's RNA code. It interferes with when the virus is performing replication, and it happens in the ribosome. So how do siRNAs work? So it finds its way into the cell, and then it goes into the ribosome, and when the virus is replicating, it inserts a part of its own code into the viruses. It replaces about 21 nucleotides into the virus and takes out that much of the viruses. And an average Ebola virus is around 2,000 nucleotides long. This means 7 codons will be different, and that means about 7 different amino acids will also be different. And it makes a huge impact. The virus essentially becomes useless and is destroyed. So in Ebola, it targets the L protein, which I'll show you a picture of later, and it causes the virus to self-destruct. And so far, siRNAs have been used to cure monkeys that have been infected with Ebola, and if these become more successful, they could be greatly used in the future for almost any kind of disease or virus. So this is a... Alright. You guys, or are you going to do it? Yes?
So where is the region Ebola is found in? Where is it common?
Africa, mostly.
Good question.
Alright, so guys, get out a piece of paper. We're going to do a quick little activity. Not hard, should be kind of fun. Alright, now write a sentence at least 15 words long, just about anything you want to. Now, everyone just pick some random word that you've heard, not necessarily in your sentence. Alright, you guys think of a word?
Yeah.
Alright, now, everyone, alright, what's your word, Zane?
Quadrilateral.
Alright, what's your word, Ian?
Stars.
What's your word, Eric?
Wayne.
What's answer a word. Kim.
Wait, just a random word?
Just a random word.
Pink.
Alright. Now rewrite this sentence, but change the 4 middle words in your sentence and replace them with those words.
What do you mean the 4 middle words?
The words that are in the middle of your 14-word sentence, cross out those 4, and just replace them with the 4 words that you have said. And then write the rest of your sentence after that. So this kind of represents what an siRNA would do to a virus. You'll probably notice that the sentence doesn't make that much sense now.
Can I read my sentence?
You go for it.
Hello, my name is a quadrilateral star lame pink of pie is apple pie.
You want to read yours, Ian?
I love how quadrilateral stars lame pink throughout your whole body without you even seeing it.
Basically, what happens to the Ebola virus, it just becomes useless and doesn't really make any sense and just kind of destroys itself. Alright. So now we're going to start some analysis questions that you guys can think about. So what do you guys think the future could hold for siRNAs?
They could cure cancer.
Cure cancer? What do you think, Ian?
They can be used for other viruses too.
What do you think some problems might be with these?
The viruses might overcome the siRNA and kind of like get immune to it, so they still keep reproducing.
Just kind of like get along with the huge mess up in the middle of their code. Now I have a quick, like, 3-question quiz just to see if you guys kind of understand.
Just kidding.
Yeah. Alright, so for the first one, where does siRNA interfere with a virus? This happens in the ribosome.
That's what I said.
Nice. And then the second part of the first one is during which phase does it interfere? And this is when it replicates, or during replication.
Is that phase 2?
Phase 2? Not necessarily. And tethrin is a cellular protein that stops viruses from getting out of cells. The third one was what does a virus need that it gets from a host cell? And that is enzymes.